Good morning, everyone. Good morning, everyone. Welcome in today's session, and let's begin with today's class. So, hope you all are doing good, and let's crack NEET PG together. So, today we are going to discuss about epigenetics. But before beginning with the topic of today, let us have a small introduction about Unacademy. Unacademy is one of the India's largest platform for the preparation of NEET PG examination, and Unacademy provides us with the best educators for the preparation of examination. So these are some of the pictures of the best educators for the preparation of NEET PG at Unacademy. In Unacademy, there is a special feature called as a special classes, and what is so special about these classes is in these classes you can directly talk to your faculty and you can get your doubts and queries solved. So it is very one-to-one kind of interaction. So special classes is a very important feature of Unacademy which is unavailable with other platforms. Then there are test series, and test series are very much important for evaluating how much you have learned so far. So for the preparation of examination, just keep on evaluating how much you have learned so far. So test series can be very much helpful in evaluating your own self. There are free mock tests, then weekly tests. So you can just go for it and prepare for the examination. Then in today's time, online learning is the best way for the preparation of examination. And why so? Because of the flexibility of the schedule. If you are a final year student or you are done with your MBBS, then if you are lacking time, you can prepare anywhere, anytime. Lectures are pre-recorded and you can just go through the lectures anytime. Then there are many faculty members who even teach in the local languages. So that is one of the additional feature that you can opt for the language of your own choice. Then unlimited practice. If a quiz is conducted, then you can go through that quiz as many number of times as you want to, and you can also watch the recorded videos as many number of times you want to. So unlimited practice is one of the additional benefit with Unacademy. Then there are many constraints in mind that if it's online teaching then it is a classroom like feeling or not. But let me make it very clear that it is almost like classroom, because when you can chat with your educator can ask you questions and you will be answering the questions, and you will be understanding that where you are getting confused, and for each and every option they'll be giving you the full explanation. So it is very much classroom like feeling. Then learn anytime, anywhere is the benefit with Unacademy. So it's just like a classroom like feeling. Then how to download the app? Just go on Play Store and type Unacademy. You will see the logo of Unacademy Learning app. Just download the app and you are good to go. So very easy for the students who are aspiring to clear NEET PG 2021. Then there is another feature of Unacademy, that is Plus platform, and in this Plus platform you will get access for all the lectures and all the topics of 19 subjects. Everything is available over there, and for any upcoming exam there are crash courses, then short term courses, quizzes and motivational videos. So with the help of those things, you can easily prepare for the examination, and it will be highly beneficial in addition of the knowledge. Then the subscription is for a minimum amount. You need to pay a very minimum amount for this subscription, and in this subscription also you can get an additional 10% discount if you are going for my referral code. So what is my personal suggestion is go for a 12 monthly or a 12 monthly or a 2 yearly program of Plus, so that you can make the most out of the available lectures on the Plus. So just go for it and prepare well and qualify the exam. And my referral code is FB01. So just by using this referral code, you will be getting an additional 10% discount in the Plus subscription. So don't forget to use my code and prepare well for the examination, right? So today we are doing a very interesting topic, that is epigenetics. So many a times students, they feel that this topic should be left out because it is quite different from the normal topic. But epigenetics is a very interesting topic if you have understood well, and very scoring as well, because the questions are like direct from epigenetics. So let us start with epigenetics. What we are going to see in epigenetics is what is the definition of epigenetics, then what is the use of epigenetics, why the nature has selected this type of nature selection, and then what types of epigenetics are there for regulation of the human phenotype. This all we are going to see in epigenetics. So what is the definition of epigenetics? Whenever there will be a change in the DNA, and that change will be chemical and reversible, and this change can be in the bases or in the histone proteins, then it will be called as epigenetics. Now, how is this epigenetics different from mutation? Isn't the term or the definition which we just saw is quite similar to mutation, isn't it? So how is it different? This is the main question. So epigenetics is reversible, most importantly. So mutation, is it reversible? Polymorphism, is it reversible? No. How is this epigenetics different from mutation? That it is reversible. It's a change in DNA. It's a change in the histone proteins also. But why it is different from mutation? Because mutation is heritable. But epigenetics is also heritable and reversible. Heritable and reversible. And when we are saying about mutation, there will be change in the bases. Here it's a chemical modification. Mutation is also a chemical modification, but there the changes, it occurs in the bases. Like a base will be changed from another base. The bases will be exchanged, or a base will be deleted out, or a base is added extra. So this kind of things all come under mutation. But when we are talking about epigenetics, it's reversible, heritable, it's chemical modification. And chemical modification of what? Chemical modification of DNA as well as the chromatin. So when we are saying chromatin, both of the things are included in chromatin. That is your DNA as well as histone proteins. And there is no alteration in the nucleotide sequence. So remember this basic of epigenetics, and you will be like have a good concept about epigenetics. How is it different from mutation? How is it different from polymorphism? So most importantly, it is reversible. Mutation is heritable, but it is non-reversible. And epigenetics is reversible and heritable both at the same time. Then the changes occur in the chromatin material and the chemical modification in the chromatin material. So chromatin, both of the things are included. The changes in DNA and the changes in histone protein, right? And there will be no alteration in the nucleotide sequence. The nucleotide sequence will remain as it is. Okay. So the term epigenetics, what does this term actually means? What is the literal meaning of epigenetics? Just now we have seen the senseful meaning of epigenetics. Now what is the literal meaning of epigenetics? Epi means above and genetics. So above genetics, which is not changing the nucleotide basis, is your epigenetics. It is above the genetics. This is one of the recently discovered method of regulation of gene expression. One of the recent method that how the gene is regulated. You know why we are saying regulation of gene expression? Let's deal this with a small example. So how to deal it with the example? Let me take a pen. So what happens is when a child is born and there is an adulthood. There is a child, there is an adult, and there is an old person. So all the three stages of a single individual, they'll be having the same genes, right? But the growth pattern in child, how the height increases in a child, like just when he's born and he's 1 year old, there will be lot of difference in the increase in height and the structure of the body. But when he's moving from his 40 years to 50 years of lifespan, there will be no change in the height. So why? Because some of the genes, they are active when he's a child and some of the genes, they are active when he is old. So different set of genes are expressed at a different stage of life. Though the whole genome is same, the genome in a particular individual, since the time there is a zygote of that individual, the genome will remain same. Now it's just so that in the embryonic stage there will be different expression in the genome. When he is a child, there will be different expression in the genome, and when he is an adult, there will be different expression, and when he is an old man, there will be a different expression. So genome is same all over the lifespan. The major difference falls in the expression of gene, which type of mRNA is being formed, and then finally which kind of protein it is forming. So this all comes under epigenetics. Now there is a associated term with epigenetics, that is your epigenome. There's another term, that is epigenome. What is the meaning of epigenome? Constellation of covalent modification of DNA and histones that impact chromatin structure and modulation of gene expression. The combination of all of the covalent modifications which occur in DNA as well as in the histone proteins comes under epigenome, right? So what are these epigenetic modifications? So we have learned the definition. Now coming to what kind of modifications are there and what is the importance. So just in previous slide we discussed that the function of this epigenetics is to express some kind of gene at a particular stage of life and to express some kind of genes in another stage of life, though the genome will remain same in an individual. So epigenetic modifications include DNA methylation and post-translational modification of histone. So basically epigenetic modification is modification of DNA and modification of histone proteins. DNA and histone proteins. So when we are talking about DNA, what kind of modification is it doing? It will be a methylation type of modification, one of them. And when we are talking about histone proteins, so when it's a protein, as soon as it is translated from mRNA, it will be showing post-translational modification. So same DNA will be forming n number of mRNAs at different stages of life, and this mRNA will be forming protein, different type of proteins at different stages of life. So this comes under epigenetic modifications. Right? Now when I was saying that what is its use, so some of the consequences and related to that, how the things they occur in epigenetic modifications. Let's start with DNA methylation and demethylation. There will be methylation of DNA and there will be demethylation as well. So how this methylation and demethylation process affect the expression, and where does this methylation is occurring? We are saying methylation occurs and methylation occurs. So which is that place where actually the methylation will occur, or is it altogether random? So no, it is not at all random process. It's a very specific process. It will occur at only a specific position in the whole genome. So DNA methylation and demethylation, it is very specific for site. Site specific. It cannot occur random. The site specific. So DNA methylation is usually restricted to CpG island. What is this CpG island? Now this CpG dinucleotide or CpG island. Let us understand this term. That which thing will be called as CpG island. So what is this? CpG. C is your base, P is your phosphodiester bond and G is your guanosine. Cytosine, phosphodiester bond and guanosine. So it will be like this kind of combination. Then again C, P and G. P, C, P and G. And in terms it will be connected with further their complementary bases, and complementary bases here also there will be a P. So like this, complementary bases. So which will come under your CP island or CPG island? This thing will come under your CPG island, and which thing is not coming under the CPG island is this thing. Like if it's a base, this is a base pair and this is your island. There is a lots of difference between base pair and island. Island will be in the single strand. The combination should be seen in single strand, and this is the combination seen in the double strand. So this is not your CpG island, but this is your CpG island. So DNA methylation, whenever it is occurring in the CpG island, what is it doing? What is its function? So its function is decrease in the gene expression. It will decrease the gene expression, and whenever there will be a decrease in gene expression or a particular gene is not at all expressing, or there is no formation of mRNA and further protein, it is called as gene silencing. So those students who are just attending the theory now, I want to tell them that I have included very important PYQs at the end of the session. So just stay tuned and just go through the lecture nicely so that you will be able to solve the PYQs. So it's a very hot topic these days for the examination. So just do not miss out anything. Right? Now we have seen that DNA methylation, it decreases the gene expression, and if there is demethylation, it will increase the rate of transcription. So how does this methylation will increase and decrease the gene expression, and which is this place where this occurs? So we will be seeing it in detail here. See, gene promoters and regulation by methylation and demethylation. So where are these CpG islands actually present? So this CpG island is present in gene promoters. It is present at the gene promoter. So whenever there will be methylation in the gene promoter, it won't let the initiation factors to localize on the promoter. And if they are localizing also, and if DNA polymerase is coming, or RNA, not DNA polymerase, RNA polymerase is coming, it won't let it slide over it. So methylation is kind of obstruction. If we are saying methylation, so what is this methyl group? It is CH3 group, right? So whenever there will be some obstruction in the promoter, like let's draw this strand and this is the promoter, and there will be extra overhangs of methyl groups. So don't you think so, if some machinery is coming over here and now it wants to slide over it, so it will create some kind of great obstruction. Like if this is our pen and there is a protrusion and I just want to slide my finger over it, if there is a protrusion, my finger won't be sliding smoothly from it. So here also, if there will be protrusion of methyl groups in the promoter, it won't let the initiation factors to localize and slide over it. So that is why it decreases the expression. So majority of the gene promoters, they have high CG content. The promoter, it contains CG content and they are methylated. So CPG islands are common sites of methylation and demethylation. So how the cell, it regulates the expression of a particular gene at a particular time, at particular point of time in the lifespan, how much it will be producing the mRNA. So that will be very nicely regulated by doing methylation and demethylation of the promoter region. If promoter region is methylated, low expression, demethylated, high expression. And now I've told you the exact mechanism that how it will stop. So hence methylation and demethylation plays an important role in regulation of gene expression. CpG island of promoters are typically unmethylated. It favors transcription. So promoters, they are usually unmethylated. If there is no methylation, unmethylated. And if it is unmethylated, it highly promotes the transcription and there will be lots of that particular type of mRNA being formed. So before moving to the another example, that is our histone modification, let's deal this thing in a little detail. Let me clear the space for writing over here. So how does this methylation and demethylation it occurs, and which are the enzymes responsible for this process? So the enzyme that is responsible for this particular type of process, its name is DNMT. DNMT. What is the full form of DNMT? It's DNA methyl transferase. So very simple, an enzyme which has got transferring activity, transferase, and the group that it is transferring, methyl group, and it is transferring this methyl group on DNA. So DNMT, DNA methyl transferase. And if we are saying there is a CpG island, so where does this methyl group is actually getting attached? Which is that place where it will be attaching itself? There must be a place. So the place where this methyl group it attaches itself is your, let me take a highlighter, it's your cytosine. This is the place where the methyl group will come and it will attach itself, and these methyl groups will be attached on the same strand of the DNA and it will affect the transcription in a lot many ways. Right, now let us move a little detail in the CpG island. Now you have understood the concept of CpG island quite well. So let's move that. Let's move in the CpG island and let's talk a little bit about this in detail. So which combination will be called as CpG island? Is a single CP and G will make island, or it should be a little big? So a single stone in a big water body cannot make an island. It should have a respectable land area for making an island, right? So same with CpG content also, that there is a specific number of CpG nucleotides which should be attached side by side for making an island. So let's talk about CpG island. CpG island. So the CpG island, the length should be more than 200 base pairs, right? I hope it's appearing. Wait, let me write it a little down because I think some of the things is getting hidden under my picture, right? So let's take a pen. Yeah, okay, now I'll write it clearly. That CpG island, the CpG island, which thing will be called as CpG island? So what should be length of CpG island? It should be greater than 200 base pairs. Greater than 200 base pairs. and GC content in this base pair should be more than 50%. Right? If it is more than 50% and it's a stretch of 200 base pairs, okay, fine, it qualifies for being a CpG island. Right? Now, this CpG island, you have understood that it is not made up of just one combination of CpG. It's a combination of 200 base pairs, and the GC percent in this 200 base pairs should be greater than 50%, should be more than 50%, or at least it should be 50%, right? Now, are all promoters made up of this CpG island, or the CpG island is the only thing which qualifies for being a promoter? So no. 60% of mammalian promoters are made up of CpG. 60% promoters. 60% promoters in mammalian cell is made up of CpG island, right? And this promoter will be definitely unmethylated here. This is unmethylated in nature, right? Now, is the CpG island present outside also, or is it present in promoter region only? So, CpG dinucleotides are really found outside the promoter region of the genome also, and when the methylation it occurs in CpG outside the promoter, it leads to CpG suppression. So what is the function? The function is regulation. The function is regulation of gene expression, right? So CpG island you have understood. Then you have understood that how much base pairs will qualify for a CpG island. Then how much percentage of CpG should be there in this stretch. And are all the promoters made up of CpG island? So no. Only 60% of all of the promoters in the mammalian cells are made up of CpG island. Now we have talked about CpG in the promoter region, one thing. Now the CpG, is it possible that it is present outside the promoter also? So there is only 4 combination of bases, right? C, G, A and T. And now they have to shuffle this thing for making n number of combination for mRNA and protein. So definitely CpG will be present outside the promoter also. So if it is present outside the promoter, what is its function? It suppresses the expression. There also it suppresses the expression, right? This is the function when CpG is present outside, and when it is present on the promoter, it won't start the transcription at all. Now let us just highlight the function of CpG island a little in detail. What is the function of CpG island? So there are 4 functions of these CpG islands. Let's just highlight it and write it in detail, that what are the functions of these CpG islands, right? So let's move with the first function. Very important, you should remember the functions well. So what is the first function? Let me take another color. The first function is cell specific expression. Cell specific expression. Like we know that all these cells, they are made up of same genome, but all cells, they are different in their morphology, in their function, in their further behavior, in their exchange of material, in their compatibility, in everything they are different. If it is belonging to a tissue, then they are different. So how is it different? How a single genome is regulating everything? So it is because of the epigenetic modification, that is your DNA methylation. Now, one of the function is cell specific expression. The another function is suppression of testis specific genes. Suppression also. And suppression in testis specific genes. Right? One function, cell specific expression. Second, suppression in testis specific genes. Now, third, control of imprinted genes. Control of imprinted genes. Fourth, X chromosome inactivation. X chromosome inactivation. So this all occurs with the help of your DNA methylation. Very important. X chromosome inactivation, is it because of your histone modification or is it because of your DNA methylation? Simple answer, one liner. It is because of X chromosome inactivation. Cell specific expression, suppressor in testis specific genes, control of imprinted genes. Very important for the exam point of view. Now we have understood everything about CG island and DNA methylation. There must be some chemistry also now behind this type of thing. So what is the chemistry behind this CG methylation? There must be enzymes like we have discussed about DNMT. So there are three variations of DNMT. They have different functions, and how this cytosine, I told you that among C and G, cytosine is changed and cytosine is methylated. So how this cytosine is methylated, on which position it is methylated, this all also one should know, right? So let's move further in DNA methylation and DNA methylation on cytosine. How does it occur? See, this is a slide of histone covalent modification. We will be using the page for understanding the DNA methylation. So DNA methylation, let's make one point clear. The enzyme that we need to use for this purpose is DNMT, that is DNA methyl transferase, right? And what does it do? It methylates the chromosome. It methylates the chromosomes. Now this DNMT, it is of three types: DNMT 1, DNMT 3A, then DNMT 3B. Three variations of DNMT. What is the function? As I told you that we need to know the functions also. So DNMT, its function is DNA methylation, right? Its major function is DNA methylation. Will directly do the methylation. Now, where will it do the methylation? There must be a place, or yeah, we have seen that CPG islands, they will be methylated. But is there any other thing also? So yes, you know about the semi-conservative model of the genetic material. The genetic material, when it is replicated, how will it be replicated? It will be like this. Let's take another color for drawing the two bands. See, one of the blue band in both of the daughter cells is belonging to the older strand, parent strand, and red one is the daughter strand. So the parent strand will be methylated. As soon as they are formed, they are methylated. This protrusion of blue color on the blue strand is telling you about the methylation. They are methylated. But the newly formed strands, they are not methylated at all. So DNA methylation on these strands is very much necessary. And when they are formed just after replication, these type of strands, what are they called? They are called as hemi-methylated, because one of these strands has got proper methylation, and the newly formed strand, it has to yet undergo the modification after replication, and they are called as hemi-methylated. So just remember this thing. So DNMT1, its function is DNA methylation, and the function of DNMT3A and 3B, both of them, they perform the function of maintenance of methylation. They'll just check whether there is a proper methylation or not proper methylation. And de novo methylation. De novo methylation. If there is no methylation signal, like when it is hemi-methylation, there will be a signal of methylating the new strand. But if there is no signal, then also de novo methylation, which enzymes will do? 3A and 3B, like when they want to suppress the expression of a particular type of gene, it will perform with the help of DNMT3A and DNMT3B, right? So you have understood about DNMT enzymes, let us move further in the chemistry, that how the things they are attached over here in your cytosine, how are they going to attach themselves. Okay, let's do it here only. Let me just erase a portion. So how these cytosines, where the methyl group will be attached, which will be that place where it will be getting attached. So let's just draw the structure of cytosine. Let's draw the structure of cytosine. So it's a single chain nucleotide. Here there will be double bond. This is NH2. This is N. Here will be double bond. Here will be your N. Here will be double bond. And here will be H. So this is how your cytosine looks like. And when there will be action of D and MT on this, what will happen, that this H will be removed with the help of DNMT enzyme, and we know which is the universal methyl group donor. The universal methyl group donor is SAM, right? So it will donate the methyl group, and methyl group will attach on the cytosine, which we are saying since long. So how the methylated cytosine will look like this, and it will be called as 5-methyl because it is getting attached at the 5th position of the cytosine. 1, 2, 3, 4 and 5 and 6. So 5-methyl cytosine. And when the SAM will donate its methyl group to the cytosine, it will get converted into SAH. What is the full form of SAH? It is S-adenosyl homocysteine. So this is a very important thing which you should remember, because it is asked in examination that when 5-methyl cytosine is formed, what is the additional compound formed? So the answer is S-adenosyl homocysteine, right? S-adenosyl homocysteine. So methyl group is transferred to the cytosine, and this is how the inactivation it occurs. And this was about your mechanism of formation of 5-methyl cytosine or methylated cytosine. Now there is another twist associated with the formation of 5-methyl cytosine. So this 5-methyl cytosine, is it a normal thing for the enzyme, for your genetic material? If it is present in your genetic material and your genetic material is undergoing repair mechanism, it won't perceive it as a natural phenomena. In some of the cases, what it does is it will convert this 5-methyl cytosine into thymine, and it can lead to mutation. So how will it form a thymine from cytosine? Let's just see the structure. See, this is your single ring structure. When we talk about thymine also, this is a single structure. And how does this happen? See, just reversal of the thing. CH3 will remain as it is. Your O will remain like this. This will be the N position, and this will be again O, and this will be NH. So this is how the reversal of thing occurs through the deamination reaction. Deamination. So there will be a removal of amine group, and it will be in the presence of water. So a thymine will be formed. Thymine is formed because of the perceivance of 5-methyl cytosine as not normal by your cell. So this creates a lot of problem, and this gets repaired, and because of the repair mechanism only there will be a change in the base. So it leads into lots of mutations, right? So we have understood this CP island in detail, DNA methylation in lots of details, right? So we know everything from the chemistry to its action. If it is not occurring inside the promoter region, it is occurring outside the region. How is it affecting? This all we have seen quite in detail now, right? Now the another thing that we need to see is your histone modification in detail. Now you have understood the chemistry. I hope everything is clear. Those who are attending the class, they can just comment in the live chat box. There is a live chat box option and they can write there and I can just take their query or their doubt or if they have understood well. So fine, let's move towards the next part of today's class. That is your histone covalent modification, right? So let me take a pen. Right. Histone covalent modification, and this histone covalent modification is also called as histone code. Why? Because now histone is regulating the expression. So histone code it is called as. So why this histone code occurs? For regulating the gene expression again. And how does it occur? So histone, it's a protein. So protein, it will undergo post-translational modification and it will do this modification. They are dynamic and they are reversible. We have seen from the very starting when we were discussing the definition of epigenetics. So we read the first word in the definition is it is reversible, heritable chemical modification. So histone covalent modification is also a kind of reversible modification. Now let us see how does it occur, where does it occur, and how will it affect the functioning of genetic material. So when we were talking about DNA modification and DNA methylation, what we said that if DNA methylation is occurring, it will lead a decrease in transcription of that particular gene. But when we are talking about histone covalent modification, what is so good about it? There will be increase in that particular gene expression. Both of the things are like quite different. So let's see how it occurs in the genome. Let's draw a double-stranded DNA. This is our double-stranded DNA. And let's take another color and draw the histone proteins. This is the histone proteins and this is how the genetic material is highly condensed in nature. It will be highly compact because of the histone modification. And we know about the charges of these DNA and histone. Our DNA is negatively charged. DNA, it contains negative charge all over, right? So it's negatively charged. And this histone protein must be positively charged for getting a proper attachment with the DNA, right? So positive, negative, they are going good together. Everything is going well. They are in a proper structure and a proper condensed form. Now, when there will be modification in the histone, which type of group is getting attached with the histones? Which group? It is acetyl group. It is acetylated. So which is this acetylated group and what are the groups? It is CH3COO, CH3COO, CH3COO and negative, negative, negative. Now your histone, it is positive and two things are negative around the histones. One of the thing is DNA and another thing is acetyl group. So now histones are acetylated. Histones are acetylated. So this acetylation, what will it do? It will lead into decondensation. Will lead the decondensation of the chromosomes. Now this decondensation, what will it lead into? It decreases the charge in histones. It decreases the charge in histones and DNA are negatively charged. Now when there will be decrease in the histone charge, chromatin will be less. Chromatin will be less condensed. It is less condensed. Now, why this less condensed is leading into higher expression? because now your promoter will be open. Promoter is open now, and if promoter is open, there will be transcription factors binding to the promoters. Transcription factors bind to promoters, and when they will bind to promoters, there will be more mRNA formation. If there is a more mRNA formation, there will be lots of gene activation, right? So this is how the whole process of histone covalent modification it occurs, right? There will be lots of gene expression. Understanding? I hope you have understood that how this histone covalent modification is performing the thing that decondensation is occurring, there is decrease in histone charge, and there will be a chromatin lesser condensation, and promoter is open for binding more of the transcription factors, right? Now coming to a little detail about histone acetylation and deacetylation. So histones, they are acetylated and they are deacetylated. So they are reversible. So definitely there will be acetylation as well as there will be deacetylation. Both of the things will occur. So acetylation, there will be joining of CH3COO group, and when there is deacetylation, there will be removal of CH3COO group in the histones, and histones, they themselves are positively charged. So histone acetylation and deacetylation is the best understood modification of histones. And acetylation, now there we saw in CpG island, where does the methyl group it attaches itself? Here it attaches itself where this acetyl group it attaches at the lysine residues. Acetylation occurs on the lysine residues in the amino terminal tails of histone protein. They occur on the tail. If it is in the interior, how is it going to affect the chemistry of bonding of your DNA and your this acetyl group? Definitely it should be on the outer environment. So this acetylation occurs on the hanging lysine residues, that is in the terminal tail of the histone molecules. So histone acetylation, if there is acetylation, there will be lesser attachment of your DNA with the histone. So there will be increase in the gene expression. There will be increase in the gene expression. And the enzyme that performs the thing is histone acetyl transferase. It acetylates the histone proteins, and if it deacetylates it, there will be more condensation and less expression. So the enzyme which is responsible for decondensation, for condensation and deacetylation, so don't get confused like the slip of tongue, that if it is deacetylation, it means it is condensation. Deacetylation is equivalent to condensation and acetylation is equivalent to decondensation. Right? Remember this thing quite very well. So histone deacetylates and it deacetylates and deacetylize and it performs the condensation of the chromatin. Now what is the mechanism of histone acetylation and deacetylation? So histone acetylation reduces the positive charge of the amino terminal tail of the histone molecules we have just read, and this decreases the binding affinity for the negatively charged DNA. And when there will be lesser binding capability of histone with the DNA, it will increase the content of euchromatin. There will be higher euchromatin. Now when the word euchromatin has come, euchromatin and heterochromatin, two words. Euchromatin means which will be forming mRNA and which will be further leading to protein formation, or euchromatin contains the coding sequences, right? And if it is heterochromatin, there will be no mRNA formation and they are non-coding. And when they are stained, heterochromatin will be darker and euchromatin will be lighter. Remember, this we have discussed in some of the previous lectures as well. So there will be increase in euchromatin formation And when there will be increase in euchromatin formation, then what will happen? More of the transcription factors can come and bind to the DNA, and when there will be more transcription factors binding to the promoters, there will be more transcription. So histone deacetylation has the opposite effect, that is decrease the transcription and increase the heterochromatin formation. So deacetylation is associated with condensation and lesser expression and higher heterochromatin formation, right? Now this another thing, that is histone methylation. One of the thing is histone acetylation. Now another thing is histone methylation, and yet another thing is histone phosphorylation. All of these things regulate the gene expression. So histone methylation and histone phosphorylation. So histone methylation, whenever there will be an addition of the methyl group, it will be called as histone methylation. And where does it occur? Occurs on the lysine residues, and very rarely it occurs on the arginine residues as well, right? It occurs on rarely arginine residues and mostly on the lysine residues, and there will be attachment of methyl group. If it is histone methylation, attachment of methyl group. The enzyme that is performing this thing is HMT, that is histone methyl transferase. When it was about the transfer of acetyl group, it was histone acetyl transferase, HAT. Here the enzyme is HMT. Now what is the function of histone methylation? It alters the chromatin configuration, which favors or decrease the transcription. It can favor as well, and it can decrease also. Both of the things can occur in the histone methylation. Now histone phosphorylation, where does it occur? It occurs on the serine residues, and there will be transfer of phosphoryl group to the serine, and the enzymes which perform this thing is kinases. Kinases, they phosphorylate the histone. And what is the functional consequence of this serine phosphorylation? Its functional consequence is, it depending on the specific residue of DNA, that may be opened up for transcription, or it can be condensed, same like histone methylation. So this is how we have seen the modifications in DNA as well as in histone proteins. Right, now let us move further in the topic of today, that is the biochemical function of epigenetic modification. What is its function? Regulation of gene or tissue specific gene expression. Regulation of tissue specific gene expression. Right, biochemical function of epigenetic modification is tissue specific gene expression and X chromosome inactivation. Right, one of the X chromosome is inactivated in female and one of it is activated. So X chromosome inactivation. Then yet another function is genome imprinting. What is genome imprinting? Gene inactivation on selected chromosomal region of autosome is called as genome imprinting. Whenever a particular gene is inactivated in a particular region of the autosome, it is called as genome imprinting. And this is the cause of preferential expression of one of the parental allele. One of the allele is dominant. So why is it dominant? Because there will be a preferential expression of that particular allele, and that we can understand with the help of genome imprinting. Remember, genome imprinting. Oh, it cannot appear like that. Just a second. Genome imprinting, right? So what are the biochemical functions of epigenetic modification? Tissue specific gene expression, X chromosome inactivation, genome imprinting. Then maternal imprinting refers to transcriptional silencing of maternal allele. If there is a silencing in the maternal allele, it is called as maternal imprinting. Paternal imprinting, if there will be inactivation or silencing of the paternal allele, it is called as paternal imprinting. Imprinting occurs in ovum or sperm before fertilization, and this imprinting is predestined even before fertilization. And this type of suppression of a particular kind of gene is leading into aging, or a particular kind of enzyme is produced in higher amount in the later stages of life. They are one of the reason of aging. How the collagen is not supported, how the melanin pigment is not supported in the further later stages of life. This all is because of change in the expression of particular gene. It is all because of the epigenetics. Right? Now coming to the therapeutic application of epigenetic modification. As I told you, it is one of the very important hot topics these days. So let us see its use in the therapeutics as well. So unlike DNA mutation, epigenetic changes are potentially reversible by drugs that inhibit DNA or histone modification. So there are various studies which reverses the aging with the help of some kind of drugs. So all those things, if there is a modification in the genetic material, just you want to remove a particular kind of group from it, so there can be drugs for changing the epigenetic modification. There is considerable interest in treating cancers that correct the epigenetic abnormality in cancer cell with some encouraging early results. Some of the genes, when they are highly aggressive in cancer cells, some of the genes which are continuously forming, constitutively they are forming, they can be suppressed with the help of epigenetic modification. Now let us go through some of the drugs which are actually doing this. So drugs that inhibit the DNA methyl transferase DNMT, some of the drugs are azacitidine, 5-azay-2-deoxycytidine and D-cytabine. Now the drugs that inhibit the histone deacetylase, or which inhibits histone deacetylases, vorinostate and valproic acid. So remember these drugs as well. Now epigenetic changes causing pathological alteration, how it leads into some kind of deformity or any kind of disorder. Let us have a look on these things. So first of it is Fragile X syndrome. Fragile X chromosome syndrome. What happens in Fragile X chromosome syndrome? The promoter site, which is actually site, will be hypermethylated, and when it is hypermethylated, so we know whenever there is methylated, the gene will be silenced. So FMR1 will be silenced, and it is the major cause of Fragile X inactivation or Fragile X syndrome. Second is cancer. DNA methylation and histone modification dictate which genes are expressed, and in turn which determines the lineage and commitment and differentiation state of both normal and neoplastic cells. So because of these changes in the methylation and changes in the methylation and acetylation of DNA as well as your histone, there are various kind of cancers. Now DNA methylation is also one kind of defense for the various kind of retroviral infections, and we have seen genome imprinting. If one of the allele is silenced, if it is maternal or if it is paternal, that is also because of your DNA methylation or histone acetylation and methylation. Now coming to some of the syndromes. First one is Prader-Willi-Angelman syndrome. The molecular basis of these two syndromes lies in genome imprinting. Prader-Willi and Angelman syndrome. Let's deal each one individually. First one is Prader-Willi syndrome. What happens in this? A gene or a set of genes on maternal chromosome, and the chromosome 15q12 is imprinted, and hence it is silenced. So When it is silent, in case of one which is coming from the mother, which one will be active? Obviously, the one which is coming from father will be active. So, functional allele from the paternal chromosome will be active. So, what are the causes of Prader-Willi syndrome? Paternal deletion of Prader-Willi locus located on the chromosome number 15 is the cause. Then maternal uniparental disomy, rarely due to imprinting defect in the paternal chromosome. These are some of the causes of Prader-Willi syndrome. Now, Angelman syndrome, a distinct gene that also maps to the same region of chromosome, that is chromosome number 15, is imprinted on the paternal chromosome. Only maternally derived allele of this gene is normal. There, only paternal derived allele was normal, and you can remember as it is P, so you can remember P for paternal activeness and P for Prader-Willi syndrome. Here, Angelman. So here in the word M is coming, alphabet M is coming, and here the maternally derived allele will be normal, right? Rest all the causes of Angelman syndrome will be same as Prader-Willi syndrome only. Now the most interesting part of today's class, that is the PYQs. Let's go ahead and solve the PYQs for what we have learned so far. Now, genes in CPG island is inactivated by what? Is it methylation? Is it methylation? Is it ubiquitination, or is it acetylation? So we know the genes in the CPG island, they are, what are they? We have seen many a times, they are methylated. They are methylated. The genes in the CPG islands, they are methylated, and specifically methylation, it occurs on the cytosine. Next question. All are true for DNA methylation except which among the following is not true? It occurs in the cytosine, so this is true. Can alter the gene expression pattern in cell. This is also true. Role in genome imprinting. True. No role in carcinogenesis, essential for normal development. So we have just now seen that it has a major role in cancer also. So everything is true regarding DNA methylation except no role in carcinogenesis, right? Let's move further in the next part. Random inactivation of X chromosome is it lionization, is it allele exclusion, is it randomization, or is it genome imprinting? So random inactivation of X chromosome is called as lionization, and it is because of genome imprinting. So here the overlapping options are lionization and genome imprinting, but you can always go for lionization because genome imprinting is the process by which it occurs, and the final result is called as lionization. Now, histone acetylation causes. When there is acetylation in the histone, what will it occur? What will it do? If it will lead into extra heterochromatin formation, increased euchromatin formation, methylation of cytine, DNA replication. So let's take a pen. When there will be increased histone acetylation, there will be heterochromatin formation. So when there will be acetylation, there will be less bonding between the DNA and the histone. So there will be increased euchromatin formation. We have seen in detail this thing also. Now, differential expression of same gene depending on the parent of origin is referred to as genome imprinting, differential expression, Prader-Willi and Angelman syndrome, genome imprinting. Now, next question. True about DNA methylation. What among the following is true about DNA methylation? Altering the gene expression. So yes, DNA methylation, it alters the gene expression. Genetic code remains intact. So yes, genetic code remains intact. Role in carcinogenesis. So yes, it has role in carcinogenesis as well. Protective mechanism against cleaving by restriction endonuclease. So there is no role in protection against the endonuclease. So what is true? It is alteration of gene expression, genetic code remains intact, and role in carcinogenesis. So for this, there are three options which are true about DNA methylation. Let's move to the next. Epigenetics is what is epigenetics? It is chemical modification of DNA. So yes, it is chemical modification. It is irreversible. No, we have seen from the very starting that it is reversible in nature. Change in nucleotide sequence. So no, there is no change in the base pair sequence. Normal variation of nucleotides. So it is chemical modification of DNA. Rest all of the things are wrong when we are talking about epigenetics. It's just opposite of rest of the options. Then methylation of cytidine residues of DNA will cause. There is methylation of cytidine residues. What will it form? What will happen? No change. There will be a change. Decrease in the gene expression. So yes, decrease in gene expression. Mutation. So yes, in many of the cases it can lead to mutation also. Increase in gene expression. So no, there will be no increase in gene expression. There will be always decrease in gene expression whenever there will be methylation of the residues. So this is it for all the PYQs, and we have learned this topic quite in detail, covering everything which could be asked in the examination. So thank you very much for patient listening, and hope you have enjoyed this topic. And if you have enjoyed the topic, please don't forget to give thumbs up, to like my video. It really motivates us. And lastly, subscribe our channel Let's Crack NEET PG and hit the bell icon for getting all of the notifications. If you are going for plus subscription, use my referral code, that is FB01. And by using my referral code, you will be getting an additional 10% discount. So let's crack NEET PG together. And this is it for today's class. Thank you very much and have a very good day. Happy studying.